My report is about a trendy anti-carcinogen target, CTLA4. I will talk about its mechanism of action, related diseases, drugs in development, and its market valuation. CTLA4 is encoded by the CTLA4 gene, which acts as an immune checkpoint and downregulates the immune response. CTLA4 and CD28 are cognate receptors expressed by CD4 and CD8 T cells. The co-stimulatory pathway mediated by CD28 activation positively regulates the immune response, while the CTLA4 protein negatively regulates the immune response. In the process of immune response, CTLA4 plays a role in downregulating T cell function through multiple pathways. In most recent research, a team at Maryland University found that the most important pathway of CTLA4 is relying on the elimination of TRK cells in tumors mediated by the constant region Fc of the antibody heavy chain and the Fc receptor of immune cells. A typical characteristic of CTLA4 gene is that it has polymorphisms. So far, people have successively discovered 4 gene polymorphisms of CTLA4. It not only negatively regulates T cell activation, but also mediates autoimmune tolerance, include T cell differentiation, and maintain the body's immune balance. Therefore, CTLA4 may be related to T cell mediated autoimmune diseases, transplant rejection, and the body's anti-tumor, anti-infection, and anti-allergic capabilities. Mutations of this gene are associated with type 1 diabetes, Graves' disease, celiac disease, etc. Next, I will talk about the drugs targeting CTLA4 in development. These are the monoclonal antibodies in clinical trials. The blue lines are clinical trials conducted in China, while the green lines are trials that are conducted overseas. First thing I want to talk about is ipilimumab, which is the world's first immune checkpoint inhibitor approved for marketing. Ipilimumab is developed by BMS. Its trade name is Yervoy. It is approved by FDA in March 2011 and approved by EMA in July 2011. It is currently the only approved CTLA4 inhibitor. At present, ipilimumab has been approved for 11 indications, 3 of which are monotherapies, and the rest 8 are combination therapies. Its indications include melanoma, renal cell carcinoma, and non-small cell lung cancer. There's some shortcomings with CTLA4 monoclonal antibodies, like its low response rate. Therefore, the more common case is that CTLA4 antibodies are combined with PD-1 antibodies. Both CTLA4 and PD-1 are of B7 receptor family. They both regulate the overlapping set of T cell signal transduction proteins in cells, but they also have some differences. When used as monotherapy in clinical studies, CTLA4 and PD-1 blockers demonstrated impressive durable response rates, increased the survival time of the responding patients significantly, and they both had a manageable safety profile. However, benefits of monotherapy were limited by low response rates. As a result, combination of CTLA4 and PD-1 blockade was thus proposed to increase the response rates and survival rates of the patients. It was thought that blockade of CTLA4, which is primarily involved in regulation of T cell activation in lymph nodes and in suppression of DC activity via TRK cells, would act synergistically with blockade of PD-1 that is mainly involved in inhibition of effector T cell and NK cell activation in peripheral tissues. Results from clinical trials that evaluated the efficacy of CTLA4 plus PD-1 blockers have demonstrated that the combination therapy can have 1 plus 1 greater than 2 effects. These are the combination therapies in clinical research. For instance, ipilimumab is often combined with nivolumab, and tremelimumab is often combined with durvalumab. The combined effect of CTLA4 antibody and PD-1 antibody has been confirmed by multiple studies. The table shows several clinical studies of durvalumab combined with tremelimumab treating lung cancer. The results of both preclinical and clinical trials have confirmed our formal statement. Combined therapy of CTLA4 such as ipilimumab and tremelimumab, especially tremelimumab, have shown good efficacy in phase 1 and phase 2 clinical studies of lung cancer, which opened a new area of tumor treatment. Clinical studies show that PD-1 plus CTLA4 can be more effective than either monotherapy. In a study, Checkmate 67, recruited patients were evenly divided into 3 groups and respectively received PD-1 antibody plus CTLA4 antibody, PD-1 alone and CTLA4 alone. A series of numbers clearly show that the combination therapy group has significant advantages. However, the combination therapy group also has some more serious side effects. In study Checkmate 214, 847 patients with untreated intermediate or high-risk advanced renal cancer are divided into 2 groups. One group was treated with PD-1 antibody nivolumab combined with CTLA4 antibody ipilimumab, and the other group received sunitinib, a currently recognized first-choice treatment for advanced renal cancer, our targeted drug. Results show that the combination therapy group reduced 32% of risk of death. The incidence of serious side effects was also lower than in the Sutent group. But more people in the combination therapy group stopped the drug because they could not tolerate the side effects. However, the combination therapies still have some shortcomings like economic burden and the poor patient compliance. For example, sometimes patients have to go to different hospitals to get injection of the 2 antibodies. Now, bispecific antibodies targeting both PD-1 and CTLA4 come into our scene. The PD-1 and CTLA4 bispecific antibody can double block the proven immune checkpoint moleculars in the tumor microenvironment, retaining the efficacy observed in the combination therapy of PD-1 and CTLA4 monoclonal antibodies, while reducing the tendency of activated T cells to attack healthy tissues at peripheral sites. It reduces the side effects of drugs and improves safety and effectiveness. Bispecific antibodies have advantages like good patient compliance, less side effects, and less economic burden. These are the PD-1 and CTLA4 bispecific antibodies in clinical research in China and overseas. From this table, we can see that the safety of bispecific antibodies is much better than that of combination therapy. The incidence of treatment-related adverse reactions is much lower. Then I want to empathetically introduce 2 bispecific antibodies developed by local pharmaceutical companies. The first one is AK104. AK104 is an internationally pioneering anti-PD-1 and CTLA4 bispecific antibody drug, independently developed and studied by Akeso Bio. It is the world's first PD-1 and CTLA4 bispecific antibody to enter clinical trials. In mid-August 2020, AK-104 was recognized as a breakthrough therapy. The FDA granted fast-track qualification for its injection. AK-104 has shown better effectiveness than monoclonal antibodies and combination therapies on treating cervical cancer and melanoma. Another drug is KN-046, which is independently developed by AlphaMap Oncology. On September 3, 2020, AlphaMap Oncology announced that the FDA has granted KN-046 an orphan drug designation. In a newly completed study, KN-046 showed pretty good efficacy in treating non-small cell lung cancer, with median overall survival of 13 months and median progression-free survival of 3.68 months. The effectiveness and safety data of the study indicate that it may become an important treatment option for patients with advanced non-small cell lung cancer. And finally is the future valuation of CTLA-4 targeting drugs in the Chinese market. According to the innovative drug valuation model of Sinolink Securities, we estimate that the CTLA-4 market is valued at approximately RMB 10 billion. Thank you for your listening.